Hello, my name is Daphne Naidoo and I work at the Center for Proteomic and Genomic Research, also known as the CPGR. The CPGR is a non-profit organization aimed to provide advanced services to the life science and biotechnology communities in South Africa and Africa. I started at the CPGR in 2019 as a bioinformatics intern, and I was subsequently employed as a bioinformatics technician in March 2020. My background is strongly rooted in biology, as I had completed my Bachelor of Science in Cellular Biology and Genetics. I thereafter went on to complete my honors in genetics. I believe that my background has played a pivotal role in my transition to bioinformatics, as it helped me understand biological problems, that being simple or complex, and the way in which those problems needed to be solved using mathematical, statistical, as well as computational tools. One of the major projects that I work on at the CPGR is the non-invasive prenatal testing, or NIPT. NIPT analyzes cell-free DNA in a pregnant woman's blood to estimate the risk of fetal chromosome abnormalities. This project is a collaboration between the CPGR, Lancet Laboratories, and Natera. Patient blood samples are received from Lancet Laboratories, and these samples are then used by the laboratory technicians at the CPGR to prepare the DNA for sequencing. Panorama is a non-invasive prenatal screening test that offers highly comprehensive and accurate screening for conditions such as trisomy 21, trisomy 18, trisomy 13, monosomy X, and triploidy. More than 2 million women in over 60 countries around the world have chosen Panorama. The reason for this could be that this test is completely harmless to the baby, as it is non-invasive. Panorama uses unique SNP-based technology to deliver the most accurate NIPT on the market. Panorama can be performed for singleton, twin, egg donor, and surrogate pregnancies, and this is the only test that is able to distinguish between maternal and fetal DNA, thus helping to avoid false positive and false negative results. Once the samples are ready for sequencing, sequence analysis is started by submitting a file containing the sample details on Constellation Portal. Sequence analysis takes between 3 to 5 hours, and Panorama starts automatically. Once Panorama is complete, JSON files are generated for those samples that have passed sequence analysis. The JSON files containing the results of each sample is moved from a Natera server to a Meditech system in order to be resulted. Once all the samples in a run have been resulted, these results are then submitted to a doctor. Bioinformatics has become an integral part in several areas of biology. In molecular biology, bioinformatic techniques is used to extract useful results from large amounts of raw data. In genetics and genomics, bioinformatics is used in sequencing and annotating genomes, as well as to observe their mutations. Bioinformatic techniques can be used to analyze and query biological databases, as well as to analyze gene expression and regulation. The field of bioinformatics is very rewarding, as the past and present scope for it increases as the need for medical science increases.